Hello, everyone, and welcome to today's webinar, Understanding Bioinformatics Workflows for NGS Analysis, Case Study with Whole-exome Sequencing Data. My name is Christy Jewell. I'm with LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Roche Sequencing and Life Science. To learn more about our sponsor, please visit sequencing.roche.com. Now, today's webinar is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. Now, I would like to introduce today's speaker, Nikita Raymond Dsouza, Bioinformatics Scientist, Roche Sequencing Life Science. For a complete biography on our speaker, please visit the Biography tab at the left of your screen. Nikita, welcome. It's so wonderful to have you today. You may now begin your presentation.
Welcome, everyone. Today, I'll be talking about the field of bioinformatics and also going over a case study explaining the steps we perform for a target enrichment analysis using the whole exome sequencing data for human genome. In our agenda, we will start out by talking about what is bioinformatics and then move to the different areas within the field of bioinformatics, followed by a case study that focuses on targeted enrichment workflow called germline variants, followed by a quick summary, and then a Q&A. So, the field of bioinformatics is complex and exciting. It spans multiple disciplines, as you see in this figure right here. And as you know, the main field integrates biology and computer science, which involves analyzing contents of DNA, RNA, proteins, etc., and trying to understand complex biological systems through these analyses. It also integrates statistics that can be used by programming tools to assess the significance of a metric, or even create a new tool where it would integrate computer science, statistics, and maths. It also encompasses biochemistry and chemistry, where we can do an in silico drug discovery using bioinformatics. And in the coming slides, we are also going to see within biology itself, there's different disciplines. In this slide, we see genomics, where it studies the structure and function of DNA sequence of an organism. Epigenomics, that studies mechanisms involved with gene expression. Transcriptomics, which studies RNA transcripts that show an overview of highly expressed or suppressed genes. And these come under next generation sequencing, or NGS, where we use the sequence of biological polymers to get insights on our complex biological systems. Next, we have the field of proteomics, where we study the protein structure and how a change in the structure of the protein can affect downstream functions. Metabolomics, studying small molecules comprising of substrates and products of metabolic processes to get insights on this biological system of ours. For this webinar, we'll be focusing on a genomic workflow that focuses further on target enrichment studies. Since whole genome sequencing is expensive and does not provide the depth of information and precision that's required to decipher the roles of individual genes or investigate rare and low frequency genetic variants, targeted next generation sequencing enables us to have this high throughput and cost effective analysis focusing on specific regions of interest. So, as you see, for target enrichment workflow, what we are going to start off is DNA extraction and then library preparation that involves a series of steps where we are doing fragmentation, end repair, A-tailing, adapter ligation, and library amplification. After which we perform target enrichment, and then these libraries are sequenced using a sequencer. And post sequencing, we analyze the data generated using open source tools. For our case, sequencing data from DNA libraries are prepared using the Kappa hyperexome V2 that is designed to capture more content from key genomic databases. We use the Avenio Edge system, which is a fully automated IVD liquid handler, and it does an end to end library preparation, target enrichment, quantification, normalization, and pooling. Once we have our three libraries prepared from the Avenio Edge system, we then move to sequencing where we use Illumina's NovaSeq and sequence our data. And then we are going to move towards the bioinformatics side, which is the target enrichment analysis. So once we have the libraries that are sequenced, Illumina creates binary base calls or BCL files that's, as the name suggests, in a binary format and represents raw data output of a sequencing run. We then perform demultiplexing, converting these BCL files to FASTQ files. FASTQ is a text based sequence file format that is generated from the BCL file, and it also stores both the raw sequence data and quality scores. Now, this falls under primary analysis for next generation sequencing. So we then perform a QC to assess sequencing quality, and if everything looks good, we move ahead to adapter trimming quality filtering. Now, in our example, downsampling was also performed to make the samples more comparable and also normalize the coverage. I have written this as optional right now, but in our case study, we do downsample to around 60 million base pairs. Next, we map these reads to a reference genome, and in this case, it's going to be AG38. This step is also known as alignment. We get a BAM file, which we then mark and remove duplicates from, and at this stage, we also calculate coverage and capture statistics like on target rate, fold 80. Now the next step will be to call genomic variants and filter them based on the quality of the variant. The whole thing here consists of secondary analysis followed by variant annotation that falls under tertiary analysis. We'll be going over these steps in details in the next few slides. And our team has also wrapped this pipeline in a container solution to help evaluate kappa target enrichment. Now, starting with demultiplexing, as you know, the BCL files are raw data generated by Illumina. So, the real-time analysis software within Illumina writes base and the confidence in the call as a quality score within these BCL files. We then use Illumina's BCL convert to demultiplex sequencing data and convert the BCL files to FASTQ files. Now, the input for BCL convert is a sample sheet, an example of which can be found within Illumina's connected software page, and the path to the BCL files. So, once we have these two information, BCL convert converts these BCL files to FASTQ files. And in our case, we have 4 lanes: lane 1, lane 2, lane 3, lane 4, where for per sample, what we do next is we concatenate or merge these files per lane and create a FASTQ file per sample. Now, FASTQ files, as I mentioned, is a text-based sequence file format that stores both raw data and the quality scores. Now, for a single run, one FASTQ file is created for each sample per flow cell lane. But for a paired end run, two FASTQ files are created for each sample for each lane. So in our example, we'll be working with paired N. So we have two FASTQ files that are created for each sample. We now move on to the next step in our workflow. Now, this is adapter trimming, quality filtering, and downsampling. Why do we perform adapter trimming? So here we have a DNA insert of size 120 base pairs with adapters on either side. And in this case, we are performing a run of 150 base pair. Now, in this case, we see this run is running into the adapter region. This is a case where adapter trimming comes into play. We trim off the excess of the adapters, and this FASTQ file would then be used for quality filtering. So quality filtering is discarding the low quality reads that have a low quality score. This is to ensure we are using good quality data for our downstream analysis. Now, FastP is used both for adapter trimming and quality filtering. Downsampling is performed using seq-tk, and as I said, in our example, we do downsample to 60 million reads because we want to make our samples more comparable and also normalize the coverage. The next step is to map the reads to a reference genome. Now in this case, it's going to be GRCH38 or AG38. We use BWA-MEM to map these reads to the reference genome and convert it to a BAM file format. So here we have our reference genome, the reads, and they're mapped to our reference genome. Now, BAM files are basically a binary file format containing all the alignment information of various sequences that are mapped against the reference sequence. We use GATK tools to clean up paired read information. So it ensures that two mates of a pair are actually in the file since we are working with paired and reads. So sometimes prior filtering may remove one mate of a pair. And here I'm showing an example of one FASTQ file that's mapped to the reference genome, but it's also going to have its other paired and read where maybe that mate might not exist. In that case, fix mate will run through the file, update the flag in the alignment file, the BAM file, if it cannot find the mate anymore. So using this information, next it's going to sort the BAM file based on chromosomes. So starting from chromosome 1, 2, and going forward. So now we have our BAM file. Next, we're going to move to mark and remove duplicates. So duplicate reads map precisely to the same location as other reads. It is important to mark these duplicate reads as they may introduce bias in your variant calling. Also, in order to eliminate overrepresentation of these sequences, duplicate reads are marked. Duplicate reads may arise through PCR or where a single library is amplified, resulting in excess of that same library. From sequencing, where sequencing in excess may result in increased duplicate reads, or RNA-seq in order to observe lowly expressed transcripts, we may oversequence high expressed transcripts, increasing levels of duplication. So a duplicate flag is added within the BAM file, and then you can either move ahead with your analysis as is by marking the duplicates, or you can remove these duplicates. Even if you don't remove the duplicates, the results do not change significantly. So once this step is completed, we then use this BAM file to calculate coverage and capture statistics. So this is the result for 3 technical replicates. we see every replicate has a 60 million reads. This is the total number of reads that we used for downsampling. Next, we move on to the duplication rate, which is low for all the samples. The mapping rate, it's 100% for all the samples, so all the reads have mapped to the reference genome. The alignment error rate, which is low. Now, this error rate is the ratio of total collected edit distance to the number of mapped bases. Next, the fraction of targets at least with at least 30x coverage depth is around 90%, which is good. Next, we have our fold 80 base penalty. So here, a lower value, as we see, indicates less variability among the coverage of individual targets. So a hypothetical case of perfect uniformity would have fold 80 base penalty of 1. So it gives you an idea about the coverage for individual targets. On target rate, that represents percent bases that fall within the target region. So here we have around 80% that falls within those target region. Now I'll walk through the variant calling and filtering process. We use deep variant, and this is a deep learning based variant caller that takes aligned reads, the BAM files, and calls variants, reporting them in a standard VCF format that I'll be showing in a bit. It detects single nucleotide variants or SNVs, and as you see here, there's a single nucleotide chain. So the sample has G, whereas the reference has A. It also detects insertions or deletions or indels where one or more nucleotide are deleted or there's additional nucleotides that are inserted within the sequence. And once deep variant generates this VCF file, we then use SNPF to annotate the variants and determining if the variants have a high, medium, or low impact, and also adding additional information within the info field of a VCF file, which we will be seeing in a bit. Now, based on these annotations that are provided by SNPF, SNPSIFT is used to filter the variants based on quality scores, the impact, and everything. So, this is the output of the variant calling and filtering. Here, it's a VCF file, and we usually parse this file if we know the list of variants we need to look at. And let me go back again. So on the top of the VCF file, it contains information of the data that the file contains, any abbreviations of those information, followed by chromosomes. And then we see a list of variants with a filter, and also provides information on genotype, read depth, allele frequency information. So what SNPF does is it adds more information in the info column, like predicting the effect of the variant, and it also adds if the variant is a high impact, medium, or low, and that's how SNPSIFT filters the variants. Next, we look at a table that shows percent of deletions, insertions, and SNVs. And in our case study, we see most of the variants are SNVs. We also have this transition to transversions ratio. For whole-exome, it is generally around 2.8. So a significant deviation from the expected value could indicate artifactual variance causing a bias. So if your TS by TV ratio is too low, your call set likely has more false positives as well. So in summary, Bioinformatics is not just computer science and biology, but can be expanded into multiple disciplines. If we go the route to understand biological polymers like DNA, RNA, protein, etc., there are plenty of tools available online that we can use to build a pipeline. Today, we did see the use of these tools and how they work in analyzing the targeted enrichment data. Targeted enrichment workflow enables sensitive and precise detection of germline SNVs or single nucleotide variants and small insertions or deletions called as indels. To make this process easier, our team has wrapped the workflow as a container based solution to streamline the bioinformatics analysis of kappa TE datasets for germline detection, variant detection. And the container includes quality filtering, trimming, optional downsampling if needed, followed by alignment, generation of TE metrics, and germline variant calling. Thank you.
And thank you, Nikita, for this informational presentation. It's been great and so much information here. It is now time for our live Q&A portion of our webinar. So to our audience, if you haven't already done so, please submit your questions in that Q&A box. Click submit, and we will answer as many of your questions as we can. Okay, Nikita, we have quite a few questions already coming in, so I'm just going to dive in here. Let's start with this one. Does the pipeline apply to only kappa target enrichment panels?
So they are open source tools, so it wouldn't be restricted to just kappa target enrichment panels. It can be used for all target enrichment panels. So yeah, so it's not just restricted.
Very good, thank you. Next question. Which of the analysis programs are free of charge?
Actually, all of them are, because they are third-party online available tools. So they're available online free of charge.
Very good, thank you. We have this submitted question. What are the system requirements for the container solution?
Okay, so like what our team has done is we've kind of built a container solution for this whole pipeline. Since we already have like open source tools, what would be required is we are using Singularity to build this whole container solution. So you would require Singularity to be downloaded and also basic command line Linux.
Very good. Let's see. Let's go with this next question. Does the pipeline work for samples and libraries with UMI tags?
So this current, like the container solution that we have and the pipeline that I've shown, it does not work for unique molecular identifiers or like UMI tags, because it doesn't take that into account for the current pipeline that I showed for whole-exome sequencing.
And Nikita, this question's come in a few different variations, but could you expand more on the transitions and the transversions?
Yeah. So transition, like the ratio is important, because there can be false positives. So transitions are basically like same nucleotide change, like a pyrimidine to pyrimidine, which is like C to T or vice versa, or a purine change where it's A to G. And transversions is changing it from like pyrimidine to purine, like A to C or G to T. And like this ratio, if it's significant, like for whole exome sequencing, it's 2.8, but if it significantly deviates from this ratio, it kind of indicates there's artifactual variance. Or if it's very low, it would indicate false positives. So that kind of, you don't know how you can trust that data to what extent as well.
So Nikita, more questions have come in regarding this. Could you please just elaborate, explain the ADBP penalty again?
So sorry, which penalty?
The ADBP?
Oh, ADBP. Okay, the fold 80 base pair penalty.
Yes.
So this kind of explains like uniformity in your coverage, and ideally it's supposed to be one. If it's higher, we do like attribute it to off targets, or like there's not a uniform coverage in those target regions, which is, I think that's how I can put it across. And that's why we look at a score which is closer to one, or if even if it's 1.5, you pretty much say that it has uniform coverage over all the targeted regions, your reads.
Very good. Are the are these tools, can they be used for bacterial genome variant detection?
Mm, they can, but like the pipeline that I showed is so I would need to take that question back as well, but I believe some of the tools can be used for bacterial genome variant detection.
Very good. What containerization and workflow management systems would you recommend?
So we are using Singularity, and that kind of helps us put the whole pipeline in one single container. And you can add, so you have your configuration file, so you can modify all the parameters that you require within this whole container solution. And then it's just one step where you just run this container, and then it runs the whole pipeline as such. Or like SnakeMake, if you want to build your own pipeline.
Very good, thank you. Next question. Hi, I understand most of the NGS bioinformatics tools need Linux-based systems. Are there any online tools available for downstream analysis?
Um, so So for Linux-based systems, I think all of them are available online. So um, you would need to download them though. I think NF Core has pipelines from Nextflow that you can use, or um, like even we could help with our pipeline where we have Singularity container solution that you can run the whole downstream analysis as well.
Very good. And we have so many questions coming in. Nikita, I want to try to combine a couple here. Let's see. Which we have lots of questions coming in saying, can you provide us with the presentation? I do want to let our audience know that today's webinar will be available for on-demand viewing. Laborats will send you an email when it's available for replay. And so you feel free to share that email with your colleagues and friends who may have missed today's live event. You'll also be able to submit questions during that on-demand period as well. Nikita, next question. Let's go with that. How do you optimize the parameters for the software?
So right now we do use default parameters, or there's like trial and error where we do change the parameters a little. But like for Whole-exome or like for target enrichment, there are default parameters that's used, and they are successful. So mostly we opt for default parameters, but if we feel like a lot of our reads are being filtered out, that's where we can change the parameter to include more reads, and not filter out that many. And yeah, that's how I would say, like, it's more of default parameters. And if we are losing out on a lot of reads, that's when we look at how we can reduce the number of reads that's being excluded as well.
Very good. Now, Nikita, we have had a lot of questions that have come in that are going to require more lengthy answers. So I want to let those audience members know that we will be answering those questions via the email address you provided at the time of registration. So Nikita, it looks like that's all the questions for today. Do you have any final comments for our audience?
Yeah, I feel like, so bioinformatics, as I said, it's like complex and exciting, and I hope this presentation helped pique your interest within it. Most of the tools are available online, and like even we do have a container based solution that we offer for like target enrichment, where it has all the steps that I've shown. And there are certain like NF Core also provides like Nextflow tools that has downstream analysis. like for this current case, we're looking at whole-exome sequencing, but there are also tools available online for RNA-seq, epigenomic studies, proteomic studies as well. So it's not restricted to whole-exome sequencing or target enrichment, but yeah, there's much more to explore. And yeah, that's about it.
Oh, very good. Nikita, thank you so much for sharing your information with us and for additionally just sharing your expertise in bioinformatics and where it fits in NGS workflows. I do want to thank LabRoots for hosting today, but additionally our sponsor, Roche Sequencing Life Science, for underwriting today's educational webcast. Before we go, I do want to thank yes.
Sorry. Thank you for having us as well. Like, I think this was it's exciting to see so many people interested in bioinformatics. So thank you for having us over.
Yes, absolutely. It is our pleasure and always our privilege to work with our friends at Roche. So I do want to thank our audience for their questions today. And as I mentioned, there were a lot of questions that we weren't able to get to today. Don't fret. Those questions will be answered via the email address you provided at the time of registration. And again, as a reminder, today's webinar will be available for on-demand viewing shortly. So watch out for that email. Come back and watch us on demand, and additionally share that email with your colleagues and your friends. So from all of us at LabRoots and Roche, please have a wonderful day. Thank you for joining us and take care.
Thanks, everyone.
Thank you. Bye-bye.